Right, I think I'm going to make a start. Welcome everyone. It's a great turnout, very exciting. First, really quickly, in the case of a fire, please make your way out the way you came in. And if you have a hearing device or hearing aid, then turn it to T, and that will improve your acoustics. Anyway, before I start, I've got a few other lectures that will be coming up, and I was asked to basically just let you know, because obviously you're quite interested in this. It's very difficult to read, but if you are interested, maybe come afterwards. There's one lecture on coral reefs in crisis on the 14th of March, and that will be in Highfield. It's between 6 and 7 in the EE Lecture Theatre. So if you maybe just Google.
There's a poster on the door outside.
Right. In that case, just basically read the information there. Next, there's also Paul Rose from the BBC. He will be in the Turner Sims on the 28th of February at 8 p.m. In that case, you would have to contact the box office, and basically the tickets will cost something. I'm not sure how much though. And then finally, there will be a Dr. Lucy Plew talking about the way people view the sea. And that will be in Turner Sims as well. And I don't have my glasses on. I can't see the girl who asked me to actually put that on. Right, the date.
It's Wednesday,
next Wednesday. And do you know what time?
It's 4 till 5, I think.
Okay, so maybe if you just basically Google Dr. Lucy Plew and then maritime archaeology, I'm sure you'll find it. Oh, and finally, I don't know, I'm sure a few of you will have already tried the YouTube link we've set up. So this is going to be our third time. We are now hopefully going to have a very good quality of this talk. And you find it at www.youtube.com/users/Nox News. And you should be able to access this talk and any future talks, hopefully, fingers crossed. And then now I've got great pleasure of introducing Ed. Ed is doing his PhD here at Nox, and he is in his 4th year, so he's almost done. And he's done his undergrad in Nox as well, but then went to go do his master in London. And he basically works in the little aquarium down, some of you might have seen earlier. But basically he's going to give a very fascinating account on why corals have colour, and I hope you'll enjoy it. Thank you.
Thanks, Claudia. Good evening, everyone. As I said, my name's Ed Smith, and today I'll be talking to you about why are corals colourful. So as Claudia said, I'm a PhD student here. Claudia? Sorry. I'm a PhD student here at the University of Southampton. I'm based here at the National Oceanography Centre. And I work in the coral reef laboratory where we grow and propagate our own corals in-house. We have a state-of-the-art molecular lab where we do a lot of our analyses. And on occasion we also get to go to the field. So to give you just a general outline of what we'll be addressing today during this talk, first of all I'll be looking at what is a coral? And really, why are they important? Why are these reefs that they build important? And then I'll move on to the pigmentation in corals. What's causing it? Does it serve some sort of function? And how can we use it maybe to tell us something about coral health? So to start with, I thought I'd go for the question, what is a coral? And I'm sure a lot of you would have played the game 20 questions. And Jenny, the first question you ask is it an animal, a mineral, or a vegetable? So I thought I'd just ask you guys to see whether you think a coral is an animal. So if you think a coral is an animal, please raise your hand. Very good. Who here thinks a coral is a mineral? Yeah. And lastly, who here thinks coral is a vegetable? Okay. Well, it's not a crazy as question as it might first sound, because it is correct that a coral is an animal. And you can see that, and in some ways you could almost think of it as, say, an upside-down jellyfish that kind of sits in this calcium carbonate skeleton. And they live in these colonies generally, although we do find some solitary ones. But like I said, they secrete this calcium carbonate skeleton. I've got a little example for you here. And these very complex structures, which you could really consider they could be acting a bit like a mineral as well. And as many of you might know, they also live in a symbiosis. So within their tissues, they actually have very tiny microscopic plants who are dinoflagellates. They're little algae, and we refer to them as the symbionts. But because they have these little plants living in symbiosis with them, and they provide a lot of the kind of energy supplies of a coral, the coral almost behaves in some ways a bit like a plant. So you see these kind of branching structures, because the coral is really trying to provide an environment where the symbionts are very comfortable and they're able to photosynthesize because the coral is benefiting from that. And to really give you an idea of how many symbionts there are, in the sort of area of your sort of thumbnail, you find several million of these little algae in that kind of area of the coral tissue, and that's why we kind of see this sort of brownish colour of corals. So why are coral reefs important? Well, I think they're important for three main reasons. First of all, ecologically they're important, and even just looking at this image here, you can see all these different species, and I had a little kind of test of myself where I was going to reveal how many different fish species I could see in this image, but actually the more you look, you start seeing tiny little fish all here and there, and I gave up in the end. But coral reefs are a really diverse environment. They've been described as the rainforests of the seas. And although they only cover less than 1% of the world's oceans, they actually make up a quarter of all the different species we find in the marine realm. Also, they're actually important economically because they provide us with a lot of goods and services. And a study about 15 years ago really looked into different environments around the world, and they were analysing ecosystems based on the services and goods that they provided us. And with the coral reefs, they sort of totted up the numbers, and they came out with a figure that coral reefs to society are worth about $300 billion a year in the goods they supply, which for me was an absolutely staggering number. And there are questions over, there might be some error in that kind of estimation, but anywhere in that order of magnitude is really, really quite significant. And lastly, socially, there are a lot of, 500 million people live kind of directly relying on these coral reefs for things like coastal protection, so they dissipate wave energy, particularly in low-lying countries. They're a good supply of food, so there's a lot of less economically developed countries which directly rely on that for their main source of protein. And obviously tourism. People like to go and see coral reefs. So they are really quite important. The problem is that they're under threat. And a study in 2008 came up with a value of about 90% of the world's reef area has already been lost, which is quite shocking. But the next fact, where within the next 15 to 20 years, they estimate that a further 15% are seriously threatened and could also be lost. And other work kind of looking into sort of the model predictions about future changes in kind of the ocean's alkalinity and temperature increases suggests that actually we could start moving from really nice, healthy, complex reefs like this to a scenario where we start to see reefs essentially that look like this. So sort of they lose this kind of three-dimensional structural complexity, and with that we'll lose a lot of the diversity of coral reefs. The various threats they're under are numerous, both man-made and sort of natural perturbations to their environment. And many of you will have seen images like this before, this phenomenon we call coral bleaching. And this is essentially where, like I said, these corals live in a symbiosis, so a relationship with these algae living in their tissues. But when they become stressed, they expel these algae, and it's really quite a remarkable sight to see a reef full of these white corals. But that's kind of my first introduction as to what coral colour can tell us, because it's already telling us here this coral isn't happy. But when this relationship breaks down, it's a real indication of stress. And we've been having these mass bleaching events which have been tied in with the increases in sea surface temperature. And this is kind of coupled with light stress, that when we get these warmer seas, then corals are more vulnerable to light, and that causes this breakdown in the relationship. But there are various other factors I've put up here things like ocean acidification, the effect of imbalanced nutrients in the water, disease, overfishing, pollution, and also as our populations increase, there's much more coastal development. And a few of these I've put down under natural threats, could also be considered human or man-made threats as well, but that's for someone else to debate. So, like I showed you with the coral bleaching, that's a very kind of simple case where coral colour can really tell us something about the reef, because if we see that, we know that these corals are under stress. So, before I thought I'd talk about coral colour, I thought I'd best introduce just generally what is colour. Now, I'm sure you've all kind of seen this experiment performed before when you were back in secondary school. It's here just to show that the white light that we see is composed of different wavelengths of light of different colours, ranging sort of from violet down here to red. And this is important to know because how a colour appears to us is it depends on how this white light interacts with the surface or solution that we're looking at. So I've got two examples here. So here's my white light coming in, but I've just represented it in the different wavelengths. And if this white light hits a green surface, the reason why this appears green to us is because this green surface is absorbing the blue light and the red light, and it's this green light that's being reflected. And again, with this purple solution, we find that when we shine white light on it, it's because it's absorbing this kind of yellowy region of the spectrum that it appears purple, because the blue and red light is being reflected. And just to really kind of give you an example of that, I've got a couple of solutions. So here is just some chlorophyll. So I extracted this from some spinach leaves from my salad, just to show you that that's a... And here we have a purple solution. This is actually a pigment from a coral. And just to show you about the differences, well, I thought, well, if I shine a blue light on them, based on the upper graph, we can see, or the upper schematic, you see that the green surface will absorb the blue light. So if I find my solution, and I shine it on here. So first of all, we've got a blue torch here. I'll try and not shine anyone's eyes. And if I pass it through the chlorophyll, you can generally see that you don't get much of this blue light coming through. Can everyone see that okay? I'll show it to those guys. However, when we look at this purple solution, which we know in theory lets the blue light through, and I shine it up, then we can see that it's letting all the blue light through. So it's transparent to the blue light, and this purple solution to us now looks blue. And that's simply because it's important that the light being shined on it only has blue light, so it's transmitting that blue light, and that's why it appears blue to us. Okay. So, now we know a little bit about colour, let's have a look at a coral and try and see what are the kind of different components of its colour. So here we have a species which is typically found sort of in the Caribbean waters, and it looks rather brown. You may say there's a sort of a slight red tinge to it, but well, we know that the brown colour of corals comes from their symbiotic algae, so these tiny little plants that live in their tissue. And it's due to their photosynthetic pigments. So, for example, in the kind of terrestrial plants we look at, most of them are green, and there's the solution from my spinach. But these algae also have photosynthetic pigments, but a slightly different composition, and that's why they appear brown rather than green. But we also find other colours in corals, and these are largely due to pigments produced by the host. And these fall into a general kind of group of proteins which are named after the kind of famous protein that was first discovered, and it was this green fluorescent protein which you might have heard of before. So they're called green fluorescent protein-like pigments, or GFP-like pigments. And they can be divided broadly into two groups. We have the fluorescent proteins and the non-fluorescent chromoproteins. And these fluorescent proteins are largely responsible for the sort of bright cyan, green, red colours that we see in corals, whereas these chromoproteins are responsible for the kind of blues and purples that we see, such as this solution here. So some of you may be thinking, well, what does he mean by fluorescence? And is that any difference to bioluminescence, another term I've heard? Well, they do have some similarities in the fact that they're both light-emitting processes, so they give out light. But the big difference between the two is that fluorescence requires light, whereas bioluminescence doesn't. Bioluminescence is caused by a chemical reaction which produces the light. And to sort of give you an example of that, here we've got a small little culture of dinoflagellates that are bioluminescent, and we're in a dark laboratory here. And just by giving the vial a little shake, you can see that these reactions are occurring and they're producing this light. Now, in contrast, if we have a look at fluorescence, I've got a little fluorescent solution here. And this is a pigment from a coral, which, if I give it a shake, it doesn't really start to glow. However, if I have a look at this, so what we'll expect to see here, let's turn this light off to darken it down, is that fluorescence requires the absorption of light, and then it re-emits at a longer wavelength. So essentially, you need to put light into the system for fluorescence to occur. So even under these kind of white light conditions, we are getting a little bit of fluorescence, but it's quite difficult to see. So one thing we can do is we can excite it with one wavelength of light, and we've got some goggles here which are just to block that wavelength of light out. And so what I'll do is to show you that if we shine the light on this protein, it's very brightly fluorescent. But it's requiring this blue light to do it. So if I move it in front of the goggles, we don't have the fluorescence. It needs that blue light. So there's absorption of blue light going on, and then it's re-emitting it as a green wavelength of light. So just to give everyone that. I have another example here of an orange one. So again, it doesn't seem very fluorescent here, but if we shine the blue light on it, then it really does start to glow. So that's the kind of the key point to really be made about these fluorescent proteins is they require some light, and it involves the absorption of some light and then changing it to another wavelength. So if we go back to our coral which we started with, and you ask the question, well, is there any fluorescence here? So what we did is I had my camera set up where I took this white light photo, I turned off all the lights, shone the blue light onto the coral, and then just put these goggles over the lens of the camera to block any of this blue light coming into my image. So all we can see is just the light that's being fluoresced, the light where the absorption has taken place, a re-emission at a longer wavelength. And what we see is really quite striking. This coral has both red and green fluorescent proteins, and where they overlap, we get this kind of yellowy orange colour. And these fluorescent proteins are really widespread throughout corals. So a report said about 97% of shallow water corals actually have these fluorescent proteins. So to give you a kind of an idea of the colour diversity of these different proteins, I thought I'd show you a few images of corals expressing some different types of fluorescent proteins. So first of all, here is a coral expressing a cyan fluorescent protein, and this is really to show you that we don't necessarily need to do this blue light setup to see them, because even under white light we're getting this fluorescence. And you can see here it's quite a cyan colour, and the fluorescence is distributed sort of fairly uniformly throughout the colony. However, when we look at another coral, we see that it's very much isolated in these tentacle tips. And again, we see that with different emission colours. So here's a red fluorescent protein, and here it's uniformly distributed throughout the coral. Whereas in this example, we really see that it's isolated in certain regions. And then we kind of come on to the fluorescent protein's ugly sister, the chromoproteins, because they don't fluoresce, maybe people have taken sort of slightly less attention to them, but they're interesting nonetheless. And we find here that they really produce these really bright, intense colours, and here we've got a nice pink purple colony. And one thing we do find about them is they generally seem to be located in the growth margins of corals. So if here's a plating coral, and these are found around the outside where the coral's growing, or in those sort of branching corals that you see in the tips of these branching corals, we do see the localization of these chromoproteins. But we don't just find these pigments in corals, we also find them in other organisms. So in the sort of closely related sort of sea anemones and cereanthids and zoanthids, we also see these fluorescent proteins and also these chromoproteins. And in some more sort of distantly related organisms, such as copepods, we still see green fluorescent proteins. And here we have a jellyfish, which is where the original green fluorescent protein was isolated from. And this green fluorescent protein that I've been talking about was one that was first discovered back in, I think, well, it was originally isolated back in the 60s, I believe. But it's taken them a while, but what they really realized with this protein was that you can actually take the gene out that expresses this protein and put it into other organisms, and we can use it to label either whole cells or aspects of cells for sort of biomedical imaging or cellular imaging. We can see different components of the cell because we label them with these fluorescent proteins. And we can do this even in live kind of tissues and cells, so we can see processes that are happening within cells in real time. And this is really quite a remarkable thing, and it's really changed the way that a lot of science has been done in this field. And as such, the original kind of pioneers of this were awarded a Nobel Prize back in 2008. And coral chromoproteins and fluorescent proteins have really contributed to this because they have, some of them have certain properties which we haven't been able to engineer ourselves. And so they really added to this, and particularly these photoconvertible ones which actually change colour, have been really important. But despite the fact they've been really widely studied in sort of biomedical science and we're really good at using these proteins, we don't actually know that much about what their role is in the actual corals. And I don't know how many of you here saw the recent BBC documentary on the Great Barrier Reef. And I had a friend text me after the presenter made a comment in that. And I put the quote up just to show you, because I think he was talking here about the fluorescence, and he says, despite lots of very clever people looking into it, no one really knows why this happens. Obviously, first of all, I'd like to thank him for his kind words and assure him the check's in the post. But I think he makes quite a good point, because really the first sort of theories about this fluorescence in corals was made back in the sort of early part of the last century, and there have been quite a lot of people really working to find out why this is happening. What sort of function does these chromoproteins and fluorescent proteins, what function do they serve? And at the moment, we don't really have a kind of conclusive answer, which is something I'm going to discuss with you and try and show you at least some aspect of the complexity of it, and to show you why maybe not so much progress has been made. So there have been various kind of suggestions as a potential function that these pigments could be serving. And today I'll largely be focusing on this first one, photoprotection, so the idea that these proteins might in some way protect their algal symbionts from excessive light. But there have been other suggestions, and the reason why I've really decided to talk about photoprotection with you rather than go into too much detail on the other ones is that photoprotection is really kind of the most hotly discussed one. It's the one that seems to be the most popular, but it really also shows the real complexity of the issue, and hopefully that will come clear in the next few slides. So the idea of photoprotection is the idea that these pigments are screening these algal plants from excessive light. Really requires a sort of a very brief understanding of light in the ocean. So here's just a graph here which shows the amount of light as we decrease in depth in the ocean. And this is in I think in the Bahamas. So coral reefs are typically very clear waters, so the light penetrates very well. And we see that in the shallow waters we have a lot of light, and as we go deeper we get less and less light. But the interesting thing here is that the kind of photosynthesis of their algal symbionts saturates somewhere around here. So at this point, any point past this sort of line, this area here, all of this is excessive light. This is too much light, more light than photosynthesis can deal with. But we also find that the majority of corals live in this kind of region. So the majority of corals are experiencing an environment where there's more light than they need. So they really have a kind of love-hate relationship with light. But the corals and the symbionts have developed mechanisms to deal with excessive light, not only with the excessive light itself, but also the consequences. So when there's too much light, we have what's called reactive oxygen species produced. And these are essentially forms of oxygen that are very damaging. And so corals have these antioxidants, which you might have heard of in your face creams and whatnot. And corals have them themselves as a way of kind of protecting them. But it was suggested that potentially these pigments might be serving a photoprotective role, so a way of reducing the light stress on these algal symbionts. And the next slide, I'm just going to give you kind of a brief sort of schematic as to how they believe these fluorescent proteins might be providing photoprotection. So on the left-hand side, we have a very basic coral, and here is the kind of coral's outer tissue layer. And down here are the algal symbionts. And on the right-hand side, we have the same setup, but in this case, we have a fluorescent protein in the tissue, whereas here it's just a transparent tissue. So if we shine a white light onto the transparent coral tissue, then all of the wavelengths pass through the tissue, and the symbionts will experience all wavelengths equally as they were in the light environment that the coral is exposed to. In the case of the coral with the green fluorescent protein, it's slightly different. As I showed you with the fluorescent protein solution, that these fluorescent proteins absorb blue light. So the idea is that they take away blue light and re-emit it as green light. So people suggested, well, we know that chlorophyll absorbs very strongly in the blue region of the spectrum. So chlorophyll absorbs very strongly blue light. So they said, well, if these proteins are absorbing the blue light, taking it away from the symbionts, and re-emitting it another colour, that maybe the corals aren't using it as much, or the symbionts aren't using it as much, then maybe it will reduce the light stress. So I've shown that kind of briefly here by removing the blue regions of the spectrum, but then we do get an extra addition of green light. So some scientists said, okay, there's an idea, but let's test it. So they went out into the Caribbean, and they said, okay, what we'll do is we'll look at sort of brown corals or corals with very low amounts of these proteins, and we'll compare them to corals with high amounts. And what we'll do is we'll take measurements of the symbionts, the little plants that live inside, and see what wavelengths most efficiently excite photosynthesis or chlorophyll. So in theory, what wavelengths are most efficient for photosynthesis? Because if these fluorescent proteins are removing the blue light, then we would expect to see the blue light to become less efficient at exciting photosynthesis in the tissue, because they're just not seeing the blue light. These host fluorescent proteins are taking it away. So first of all, they perform the measurements with a brown coral, and essentially here we have the different wavelengths, the different colours of light, and on the y-axis we have essentially the most efficient wavelengths for photosynthesis. So the higher it is, the more efficient it is for photosynthesis. So here in the blue region, we see this kind of peak of the wavelengths of light most efficient for photosynthesis. So when they compared it to a green coral, the best example they could find of any kind of removal effect of these green fluorescent proteins was this coral here. And this coral has a protein which absorbs very strongly in this region of the spectrum, and we see a decrease that corresponds to it. But they kind of said, well, the difference between these two is about 10%, which, if you think about your kind of sunscreens that you put on at the beach, would probably equate to maybe a factor 2. So it's really not very effective. So they said, well, if this is the best example we can find, it's probably unlikely that they're performing a photoprotective function. And that's really kind of the point they got to, and there still remains a lot of kind of debate within the scientific community, but that's the real kind of sticking point at the moment. So there still are some arguments for and against. So we know that these proteins are light regulated, so if there's more light, we'll find more of these proteins expressed. The absorption bands, a lot of these green fluorescent proteins absorb in the blue region of the spectrum, so ideally the most strongly absorbing wavelength for the chlorophyll. So they say, okay, they match up. So again, if you were to have a photoprotectant, that's a good place to have it. And lastly, some of the corals they looked at, which had the fluorescent proteins, are less susceptible to light stress, which again indicates that maybe it does have something to do with light stress and removing that. However, the arguments against, so the graph I just showed you, they're called action spectra, and they showed very little or no evidence of any kind of removal of light from the symbionts. And in the same study, they tried to see a trend with depth, because you'd expect that maybe in the shallow water you'd have a lot more where there's more light than in the deeper waters, but they couldn't find a trend. And lastly, and again, this is why I showed you the slide to show that these fluorescent proteins are found in other organisms, because you also find fluorescent proteins in species from the deep sea and also in species which don't have symbionts. So it kind of raises some questions, and here's just an example of a stereanthid from, this is in feet by the way, so 1,740 feet deep. So there's definitely not going to be any real kind of light stress. So why would you need to be expressing these fluorescent proteins? It's a complex question. But we move on. So we move on now to these chromoproteins, so the ones that aren't fluorescent. And these are kind of been slightly ignored because, like I said, they're sort of the ugly sister. But actually, they make things a lot simpler for us, because unlike the fluorescent proteins which absorb and then re-emit, these ones simply just absorb light, and they absorb light very strongly. So in this case, if we shine white light onto this tissue with these chromoproteins, we'll find that it should remove kind of yellowish wavelengths of light, and so therefore their symbionts should be less efficient at using yellow light. So we went out to replicate the study from the Caribbean and measured some brown corals, and you see a very similar trend. However, when we looked at the corals with the chromoproteins, we see this big decrease, and at the kind of peak it's about 50% less efficient at using these yellow wavelengths of light than the brown coral. So that says, well, maybe that is photoprotection. And really just to see that it is due to these proteins, if we have a look at the absorption of these proteins, they absorb very strongly in this yellow region, which is where we also see the maximum difference. So again, at this point here, it's about 50%, and if we kind of equate it over this whole spectrum, it equates to about somewhere up to 10% of the total light field that these symbionts experience is reduced. So it's certainly no kind of factor 50, but it's about a factor 15 if you think about it in terms of your sunscreen. So it's doing something, and it does really give us a nice clear indication that these chromoproteins are acting as photoprotectants. But like the fluorescent proteins, there's still some debate going on as to reasons for and against. So reasons for, we have this action spectrum, so the graphs I just showed you do seem to support the idea that they might be acting as photoprotectants. They also have this light regulated expression, and although it's not been looked into in too much detail, there does also seem to be a distribution with depth. But we also have some sort of questions. So first of all, they have absorption outside the major pigment bands. So like I said earlier, if you want to build a perfect photoprotectant in the visible wavelengths for a coral, you probably think about one which absorbs blue light. But one thing we do know is that a lot of coral processes are regulated by blue light. So actually it might be counterproductive to actually have something which is removing that blue light from your other tissues, because you're then messing up your control mechanisms. And also, as I mentioned earlier, we have localization in some species of these chromoproteins in the growth zones. So here's just an example of a branching coral. You can see that in these tips, and I've just enlarged one here, we have a very high concentration of these chromoproteins. And some of you, quite rightly, might be asking, well, what's the point of having a very high concentration of these chromoproteins in a region where there's no symbionts? So you can see these kind of brown symbionts here, but in a lot of the tissue, there's not many symbionts. But there have been some modelling studies on this, and actually what they've shown is that the coral skeleton is very, very reflective, and it traps light inside. So the light field inside the tissue is very, very strong. And so by producing these chromoproteins, they reduce the light stress in these environments and allow the symbionts to establish their population within these tissues. So that's kind of an overview of the potential functions of these proteins. Now it's probably important to ask, well, what can it do for us, or in some ways, what can it do for the reefs? And using coral colour to monitor coral reefs, particularly these reefs under threat, there's been something that's been done for probably the last 10, 15 years, but it's certainly not an easy job. So we can see coral reefs from satellite sensors as well as airborne sensors, and this is just an image from an airborne sensor. But the great advantage about monitoring coral reefs from these different kind of sensors is we can cover such a large area. So if you look at the scale bar here, this is 200 meters, just this distance. If you imagine how long it would take a diver or a team of divers to cover and map this whole area, it's an absolutely astronomical amount of time. And this is just simply an airborne sensor which covers a very small area. If you go to a satellite sensor, you can cover vast regions of reefs very easily. But you need to know about coral colour in order to be able to interpret these images, because the way that corals absorb and reflect light is really important in understanding and being able to pick out, say at this point here, what is actually under this water. And by knowing how corals absorb and reflect light, but also knowing how the water column absorbs or attenuates light is very important. And here's just an example of so what they've done is applied kind of a model to remove the effects of water, and then it makes it a lot easier to interpret. But it's just really to emphasize that you need to know something about coral colour in order to interpret this. But the real kind of goal, the real thing that we're trying to do is not just to be able to say, right, we have this area, we can say there's some coral here. We really like to know, well, is this coral healthy? Is there some way of, some sort of indication that this coral is under stress? And it's quite difficult still to really pick up, even comparing dead versus living corals using satellite methods, which is somewhere where these fluorescent proteins might come in to help us, because these fluorescent proteins are only localized in living tissue. And we also know under certain environmental conditions, sometimes the coral will stop producing them, and other times it will enhance its production of these fluorescent proteins. So these could really tell us something about the reef, and we can use these in reef monitoring. And really just to kind of sort of finish the talk and to give you an overview of sort of how diverse and how widespread these fluorescent proteins are, I thought I'd show you a little video from when we were out in the field last year. And this was simply taken with us snorkeling on top of the surface of the water and using these blue torches and yellow filters to see the fluorescence on the reef. And if anyone here suffers from motion sickness because we're on the surface and it was quite choppy on that day, you might want to look away. But as you can see here, like as we move along, we can see the different patterns of reef fluorescence, and there's an absolutely enormous amount of fluorescent corals here. Now, some of you might be thinking, well, do all corals have this fluorescence, or is it a way of monitoring all corals or just some corals? Well, we know that about 97% of shallow water corals do have this fluorescence. So it really should be a tool which is really quite useful in the shallow waters, but it's something that really needs to be evaluated a lot further before we can really start using it sort of operationally. But there's a lot of promise in this as a sort of a technique in really trying to understand the health of the reefs, which is quite important when we see all the threats that coral reefs are facing at the moment. So, just to summarise the talk, I thought I'd go back over some of the questions that I posed on the initial slide. So first of all, I think we can see here that corals are colourful, but the question of what makes a coral colourful, well, I think we've learned today that it's a combination of the algal symbionts that live in the tissue, as well as the host-produced pigments, so these fluorescent and non-fluorescent chromoproteins. The title of the talk was Why Are Corals Colourful? And I feel in some ways a slight fraud because I haven't been able to give you a kind of perfect answer, but hopefully I've given you a little bit of an insight into the complexity of trying to work out what the functions these are. But I think at least with the chromoproteins, I think we can be fairly confident now to say I think they are acting as photoprotectants. But at least with the fluorescent proteins, it's certainly something where more work is going to be done. But the fluorescent proteins in particular do give us a very good opportunity of monitoring coral reefs and in a new kind of shed some new light on the processes that are going on, because coral reefs are under threat and they really need our protection. I'd like to thank my colleagues in the Coral Reef Lab, Martha, for letting me have a look and film her bioluminescent cultures and bifungus, and ask if there are any questions. Thank you.
Thank you very much. That was brilliant. Do we have any questions for Ed? Don't be shy. If there are questions in the seminar room, please walk up to the lectern and use the mic. I have just turned it on, so you should be able to ask us something from in there, if you want to. You don't have to. Yep.
Perhaps a slightly embarrassing question about photosynthesis. I thought photosynthesis was just for plants. You started off by saying obviously corals are animals.
Yeah.
Can you just explain exactly why you want to be protected from or develop photosynthesis and make sure because that's going to be the essence of why you thought photoprotection is the reason.
Okay, so the question is how does kind of photosynthesis relate to a coral? Why is coral really worried about light? And the real kind of key thing here is the symbiosis, so the relationship between the coral and the algae that live in its tissues. And the coral seems to be very sensitive to how the algae are and what kind of state they're in, because essentially what happens is that when the algae get stressed, they start to almost become, the relationship breaks down. It's kind of, if anyone's in a relationship and things, we have some rocky times and a few troubles, and that seems to be what happens. And it's the symbionts are, well, some are a lot more sensitive than others. So again, it's like having a partner. Some are more sort of sensitive about this. But I'm trying to word this, put this very sort of subtly. But it's the case that the coral, in some respects, really does behave a lot like a plant because it's so sensitive to the algae that live inside its tissues, because it really does benefit so much from having them there. And that's why the corals become so vulnerable when this relationship breaks down, because they're so heavily dependent on having these symbionts that when it breaks down, then the coral's in trouble, really.
When you are showing us the slides, when you're doing different lights onto the different colours of corals, surely they would be getting white light.
Well,
because the light's being filtered by the sea.
Thank you.
Yes. So the question was, corals would very rarely receive white light because in the underwater environment, water is attenuated, so you get a different spectral composition. So yeah, I used here a white light example, particularly for the chromoproteins because we largely find them only in the shallowest waters. So the solar spectrum in particular is very strong in yellow light, and in the first, well, even at I think 3 meters depth, there's still more light at the yellow wavelength than there is even in the blue. But as you get deeper, you lose red light very, very quickly. And also the yellow light decreases quite a lot. So this really kind of points out why seeing a distribution with depth with these chromoproteins is also important, because they're only useful in the very shallowest of waters. And again, this is there's some more questions in terms of, I put up one of the potential functions as photo enhancement, because there'd been a suggestion that maybe in the deeper waters you have certain fluorescent proteins which provide a wavelength of light which isn't present at that depth, and maybe in some ways the algae might need that extra wavelength of light which they're not receiving at those depths, and so it's a coral's way of kind of enhancing photosynthesis.
Any further questions? Yep, at the back.
Well, structurally and chemically, what actually causes them to be coloured? These proteins.
So the question was, what kind of sort of chemically or structurally provides the colour in these proteins? And well, the thing about these proteins is they have a kind of a very sort of unique and structure where it's like a protein which is almost shaped like a barrel. And in the very centre of this barrel is a sort of a 3 amino acid sequence which forms what we call the chromophore. And this is the part of the molecule which is responsible for the colour. And essentially the amino acids that make up that chromophore and also the orientation of it are what cause the different wavelengths that we see. So whether one of these proteins is fluorescent or non-fluorescent, and also what wavelengths of light it absorbs and emits depends on the makeup of this chromophore. But it does kind of bring in a new point that, like I said, it's a protein which is a big kind of barrel of a protein. It's only this very small part which is colourful. And you may argue that maybe we look at the colour because that's the thing that sticks out to us, but there could be something else about this whole protein which could be doing a function which is completely unrelated to light. There could be a binding site or something like that going on with this protein, and so that could be involved in the function instead.
Any further questions? Are you sure? Oh yeah, another one?
Have you done any analysis of frequencies outside light frequencies? I mean, either infrared or ultraviolet, and are there any interesting elements beyond the visible spectrum?
So the ultraviolet is very interesting, but it's one because some of these proteins do have absorption in the ultraviolet, but again, using those actions, but it doesn't seem to be having an effect. Now, the infrared, at the moment, we haven't found any which are involved in any lights near those wavelengths, but there are a lot of people who would like to find one, because the thing with this cellular imaging is that the longer the wavelength you get, the less kind of damaging the excitation light will be. So if you could find one of these fluorescent proteins which emits light in the infrared, then it would make using these fluorescent proteins a lot easier and a lot less damaging to use it on live specimens. So that's really kind of almost the holy grail, but I'm not sure whether it's possible to find it or not.
Yep.
On land, you have animals that see colours differently to how we see them, and a lot of pollination and so forth depends on how the insects see the plant in the way that they see it as opposed to how we see it. Is there any possible correlation between possibly even the symbiotes are reliant on the coloration to help them in that way, rather than obviously corals being animal or mainly animal?
So the question was whether the kind of visual systems of other organisms are important in terms of the emissions of light or reflectance of light by the corals. And it's something that has been looked into, maybe not in depth enough, but there was a suggestion that potentially by having these fluorescent proteins, they could mask the appearance of the symbionts within the tissue. So the idea is that by having kind of a colour screen, so no longer would your tissue look brown. So if you're a herbivorous fish looking for something to eat, then by covering up the brown, the fish will swim by rather than try to get at the symbionts. And that was done sort of using models of fish optical systems. But again, like you said, colour is really kind of an important thing ecologically speaking, and I don't think there's been enough work done in that kind of respect, because there's also settlement cues by coral larvae seem to have certain colour preferences as well, which could also be related.
Any more questions? None? Oh, yeah.
So the fluorescent proteins are distributed outside the fleshy parts of the polyp, so within the calcium carbonate skeleton. Are they secreted extracellularly? Are they always within cellular strip?
So they're always in the tissues generally. So corals, some kind of related corals have pigments in their skeleton, but these specific types of pigments are only ever found in tissues. Now, how they're distributed within the tissue, sometimes they're in small little granules or like sort of aggregates of these proteins, and other times they're evenly distributed in the tissue. But yeah, we always find them within living tissue. So when a coral dies and the tissue goes, we can tell that because there's no fluorescent signal.
Any more questions from this room? No? Any questions from next door? No one wants to go up to the mic? No. Okay, in that case, we will say thank you very much to Ed.
One thing I'd just like to say is that if any of you are on Facebook, we've just set up our Coral Reef Lab Facebook page, just so we can give you a bit of information about what we're doing and maybe future talks that we might be having, or maybe even if there's an interest in it, we could do some little tours to see the fluorescence in our aquarium. So if any of you are interested, I think the name of the group is Coral Reef Lab NOC in Facebook. So if you want to sign up to that, and just let us know.
And before you all run away, I'd like to say that next month's talk is by Charlie Main, and it's talking about the ecosystem effects of deep sea oil blowouts, so things like the deep horizon, and maybe how that might have impacted the environment. If you would like to come along next month. If you want to see any of the talks that you've missed this year, they are online. I'm sure Claudia mentioned this. The link at the top, it is a link we'll be adding to all our advertising, leaflets, websites, etc., so you'll be seeing it a lot. And thank you very much. Look forward to seeing you next month.